This Week in Microbiology is brought to you by microbe.tv and is supported by the American Society for Microbiology. To learn more about microbes, visit microbe.tv and asm.org. This is TWiM, This Week in Microbiology, episode 332, recorded on May 15th, 2025. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michelle Swanson.
Hello, and it is a beautiful day. It's bare arms and bare legs kind of weather.
So it's sunny.
Yeah, and warm, and things are in bloom. It's lovely.
Here it's been raining all week, and today it's raining and chilly. But they tell me life needs water, so
it does.
Here's the water. Here's the water. Very good. Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. And we do indeed have spring. It's not moved into summer yet, thankfully. And it's, as they say in the press, Chamber of Commerce weather.
Chamber of Commerce. If you enjoy these programs, these science programs, we bring you real science, not misinformation, not lies, then we'd love your support because it costs money to make them. We do not have advertising on our programs. I don't like to interrupt science with ads. And your little donations here and there help us. Microbe.tv/contribute. All right, today, a couple of very interesting stories for you. And the first I will do as a snippet. The International Space Station has a unique and extreme microbial and chemical environment driven by use patterns. This is in the journal Cell. We have two co-first authors, Rodolfo Salido and Haoshi Nina Zhao. And the last author is Rob Knight, who has been on TWiM before. So this is from a variety of many different places. Looks like mostly in California. We have University of California, San Diego. We have Caltech. We have the Johnson Space Center in Houston, which, of course, if you're going to do something with the space station, you have to involve them, of course. And
people from outer space.
People from outer space who
collected the samples.
They would have to collect them because Rob Knight cannot go up, or his postdoc cannot go up.
No.
There are also contributions from Caltech and University of Denver.
Yes. After you hear this, you're not going to want to go into space, in my opinion.
And
I think people who go into space are very brave, and people who are volunteering to go to Mars are even braver, of course. But
or dare you say crazy?
I wouldn't say crazy because we need brave people to push the
to go exploring. That is true.
Frontiers, and that's always been the case. But this International Space Station, which is composed of modules from different countries, it's a built environment, right? It's built by humans. It's not like a tree or soil or oceans. That's what we mean by built environment. It's completely isolated from Earth. It's existing up in space, and then there's a little bit of traffic back and forth. And so it's important to have healthy astronauts, right? So you really have to understand the environment, and particularly in this paper, the microbial environment and how it relates to Earth, right? Is it different from Earth? Is it similar or whatever? Because we know on Earth humans do pretty well. And it turns out from this paper that it's very different and it needs improvement, right? So as we have evolved on Earth, we shape our environment, we shape our microbial communities, among others, we shape our chemical environment, and presumably the same thing happens in space as well.
And they shape us.
And they shape us. For sure. Now, they tell us in the introduction that space flight can detrimentally affect the immune functions of space crews. and you can have antimicrobial resistance, you can have biofilm formation, you can have virulent microbes. And to try and control this risk, the space agencies survey the microorganisms that are on these, not just the U.S. but other countries as well. We had recently Mark Martin talking about the China space station and a microbe there. Um, Now we've done already a lot of work on this. This is quite a pioneering work that has some new approaches, but it's been done before. There have been culture independent studies where you swab and you sequence, and they have shown that the space station is dominated by human associated microbes that change as crews change. Right, that makes perfect sense. But these studies have also shown that between the ISS and the ground, the built environments select for extreme physical and chemical tolerance. So we need to know more about the potential threats uh, in these built environments in space. And one of the things they point out is that there are no beneficial microbes in the International Space Station.
Well, if I could just point out why this matters to the general public
Mm,
is because the built environment in hospitals is very
yeah.
similar to the space station. We have a lot of plastic in hospitals. We
Mm.
have controlled atmosphere in hospitals. We control the humidity in the hospital like we do in space. And we have unique microbes in the hospital and unique circumstances. We may not have the cosmic rays that the space station has, but we do have multi drug resistance selection pressures being put on the patients, the healthcare workers, and the microbes. So, understanding what drives the data that Vincent's going to reveal to us can have real, significant, life altering effects here on little old planet Earth.
So they go on to tell us that on the space station, astronauts get a number of ailments like rashes, atypical allergies, immune dysfunction, including reactivation of latent herpes viruses, fungal infections, bacterial infections. Sorry, folks, enough for me. I'm not going there.
You're not going to space.
Not that anybody ever invited me, and I'm too old anyway, but
And there's not a hospital nearby.
there's no hospital. And if you need to get back to Earth,
Yeah.
it's not trivial, right?
No pharmacy nearby. Yeah.
You can't call Uber and so forth. All right. So here in this paper, they want to have a more detailed look at the microbes and also the chemicals on the space station. And they have a three-dimensional microbial mapping survey, which they call 3DMM, where they can map where in the space station the microbes that they find are. And they're focusing on the U.S. portion of the space station. It's called the U.S. orbital segment. And they compare what they find on that with what's on the ground. All right, so we have a huge database of what's on the ground, and obviously this requires a lot of computational power and the ability to write algorithms that can compare them. So that's what they're going to do here. So they have 803 environmental surface samples. This is Expedition 64, 737 surface swabs, and 66 controls. And they got, these modules in the ISS have different levels of occupancy, and they got more samples from those with higher occupancy. They were sequenced, and then the data were visualized through this three-dimensional model of the space station, which is very cool, right? You can see what's on all the surfaces. And they identify the species. They're able to identify 440 and 896 species using two different databases. And all these data are available. They give a website where you can go to and check it out. They have both the identification of the microbes. They have the genome sequences. They didn't do full genomes for all of them. Most of this is based on 16S ribosomal RNA sequencing. And then the mass spectrometry to analyze the chemicals that are there.
While you're catching your breath, let me just add one more fun fact for our listeners. The average human loses between 40 to 50 million skin cells per day. And remember, they all have a skin microbiome affiliated with them. So that's a lot of dead skin coming off of the astronauts, 40 to 50 million per day.
Yeah, in a closed environment, right?
In a closed environment.
So here on Earth, it just becomes dust.
Yes.
Ashes to ashes, dust to dust, right?
So you're telling me that astronauts aren't wearing their big balloon suits that we think of when we think of an astronaut?
There are no Michelin men in space.
No, you take it off when you get in there. Yeah, they're just wearing, whatever the outfit is. So they sample 9 modules. They have similar atmospheric conditions. They want to know if the diversities vary among modules, and they find yes that it does. It depends which module it comes from. It influences the diversity, the kinds of species that you find, and use patterns influence that. So, for example, one of the modules, the Japanese, the module where they do personal hygiene in the Japanese side, like body washing, face washing, and teeth brushing, is going to have one kind of microbe, whereas there's another module devoted to experimental activities that has a different kind of genome, of species of bacteria. So what's the origin of these surface microbes? Remember, the space station has been away from Earth for 2 decades, right? It was launched 20 years ago. And so what are the contributions to the microbiome? Well, building materials, crew member rotations, which we've talked about, resupply missions, including food supplies for maintenance, experiments, everything's brought to the ship is a source of microbes. And also they reconfigure parts of it, and that can cause shifts in the microbiome. And so what they did was to focus on human host associated samples. Also, human skin, oral cavity, nose, feces, and urine, and building materials. And then the free living environmentally associated microbes were represented by samples from terrestrial soil and water. Okay. So, as they say, so the ISS, again, is a built environment. And like other built environments, human skin is the primary source of microbes in the International Space Station. And contributions from free living terrestrial sources are minimal, as you might expect. And next, building materials contributed the most microbes. They also find that module use, as I've already mentioned, influences the contributions. Like there are areas for dining, food preparation, and food storage. And guess what? Food microbes predominate in those areas.
Reassuring.
Now, here's the thing that really impacted me. So there's a compartment for waste and hygiene, right? It's a bathroom. And you can't flush the toilet. You have to store it. And so just by swabbing, they get fecal bacteria, and you'll see later, they get chemicals from urine. So do you get the picture? It's floating all around.
We have a term for that in infection control in the hospital. That's referred to as a fugitive emission.
Fugitive emission. It happens on Earth too, eh?
But let's not assume they're bad. They're all bad. There might be some good ones.
They're not all bad.
It's just, it's the yuck factor, Michelle, right?
Sure.
It's a little weird. Weird.
Not all fugitives are bad. It's
well,
just
and we're going to hear more about that
escape.
from Michael's paper. There are some good ones that you want in there.
By the way, the most abundant ISS microbe was Staphylococcus.
Not surprising. It's affiliated with the skin.
Right? Yep. So from skin and nose samples. And so, all right. So next, some chemicals, and we'll get back to bacteria. They use mass spec to try and identify chemicals that are there. They're captured on sources, and they can sort of tell where they come from. They compare the mass spec profiles with things that we've done here on Earth, and they can identify things. And they say it's interesting, a large portion of these chemicals, surface chemicals, probably come from microbial sources, right? Followed by food sources, human-derived sources, or this is a very interesting one, personal care products. So they use personal care products so that they don't dry out their skin, right? They have to remain supple, and so they get, you put some hand cream on, then you go to touch an instrument, and it gets on the surface, and then you come in and swab it and you measure it. So
the microbes will eat that hand cream because it's got all sorts of good fat in it.
Yep. And then you swab it and you get it in your signature. So basically, they find that the module use drives the surface chemistry, right? Same as before. So I'll give you some examples, some hotspots. They have an exercise device. It's called the Advanced Resistive Exercise Device. They have an oxygen generation system, and the toilet, I'll call it. They have 3 distinct chemical hotspots. This exercise thing has its own chemical signature. It's got corrosion inhibitors and sealants, rubber additive. I think those are parts of the machine, right? In the first place. The toilet, they call it the space toilet, had the highest abundance of urinary metabolites. And they list them all. So that's what I'm saying. It's very difficult to contain this in the ISS. Also, chemical hotspots at the dining table and food storage rack. It's probably the same at home, right?
Sure.
You wipe the surface, you're going to find these chemicals as well. And there are module-specific aspects of the chemical variation, also the chemicals that they find. They also note that disinfection activity in one of the modules influences the chemical profiles on the surfaces, as you might expect, right? Because you disinfect and you're going to leave behind chemicals. They say also influences the microbial life there. If you disinfect, obviously, you're going to get rid of them. And they said disinfection was discouraged for a minimum of 4 days before surface sampling, but still they found an effect of disinfection activities on the surfaces. So what about comparing the ISS with other built environments? Because, other built environments, as we said, I like this sentence they write, modern urban living significantly reduces exposure to environmental microbes and increases exposure to human-made chemicals. So even here on Earth, I don't know what to do. Live in a cave?
Well, and there are studies that show that people who grow up on farms are less apt to have asthma and things like that because they've had healthy exposures.
Right. Well, I didn't grow up on a farm. I don't have asthma. Do you have asthma, guys?
Nope.
Did you grow up on a farm?
Nope.
No, but I'm sure I'm inhaling some dirt every now and then.
You were inhaling dirt. Okay.
We, from the age of 3, we were thrown outside and played in the dirt. We had a sandbox.
Dirt under your fingernails.
Yeah. I played outside all the time. Yeah.
Yeah. It was a very less structured play. You, as the child, made your own entertainment. And we'd get home from school. We'd go out and ride our bikes and go to the ball field, play ball, play football.
And Vincent, probably riding the subway in New York, you're getting some pretty good exposures.
So they do, what they do is basically compare their sequences on the spaceship with those on Earth and in a range of settings. They have a couple of different categories of settings, like homes across urbanization gradients in different countries, before and after human occupancy, confined human spaces, closed habitats, offices, indoor construction materials. So you get the idea, right? We have a lot of data for that on Earth, and they use their software to compare microbial composition across these samples. And I've said before, they find that the space environment is similar to built environments on Earth, but more extreme. It's the most extreme of the built environments because it's up there and isolated. For example, Staphylococcus, the dominant bacterium up in the space station, is also dominant in built environments here on Earth because, right, Michael, the skin is shedding and it's inside.
And you also have to appreciate that the signature, what most people think of Staphylococcus, Staph aureus, that last species name, aureus, means it produces a golden pigment. And microbes produce pigments to effectively deal with the insult of radiation, whether it be UV or cosmic and so I was interested to see if they talked about pigment producing microbes to see if the Staphylococci they were recovering were more pigmented, but they didn't grow them. So absence of growing, you wouldn't necessarily know if they were producing pigments, and pigment expression is often determined on circumstance of what selective pressure is turning on those genes, et cetera.
So along with the high amounts of Staphylococcus, there are reduced numbers of other bacteria like Chitinophagiaceae, Penibacilliaceae, Burkholderia. And they say that the dominance of staph is different in environmentally exposed built environments. In other words, if you have windows and doors that you can open, right, you get a contribution. Well, the
those
skin
organisms
goes outside,
are soil,
and those are soil organisms.
It's coming in on the dust,
and the dust, your skin can float outside. So they say maybe we could balance this spaceship. Maybe we can introduce a non-saline soil matrix or something somehow in built environments like the ISS to more align it with communities found in environmentally exposed habitats, right? And you could experiment with that on the ground, of course, make sure it's safe, of course. And as far as the chemicals, the molecules shared across the whole urbanization gradient that I've just mentioned before include terpenoids and fatty acids, whereas in the ISS, the families shared between the ISS and city or townhomes are characterized by synthetic chemicals like polyethylene glycol, quaternary ammonium disinfectants, and the personal care stuff. And so they say this further emphasizes the extreme nature of the ISS in terms of built environments. They also do some whole genome sequencing, and they want to in part look at antimicrobial resistance genes. And so they look for those and they find 1,065 AMR genes in their sample of 22 different classes, and they're present in more than half of these are present in over 90% of surface samples. So a lot of antimicrobial resistance. I don't know how that would compare to Earth, Michael. What do you think? Would it also be high or not so high?
In hospitals, it's really off the chart. But when you go to your average house, it's not.
Okay.
And it's again because we're not living under the selective pressure, which because antibiotic resistance traits carry a cost. And you need to keep those genes around if you're being continuously threatened with an antimicrobial, whereas at the house, you're not. Yeah. And so I think these microbes in space, because of the widespread use of the harsh chemicals to keep things sanitary, that may be, and a lot of the AMR genes are on same operons as many of the genes that degrade disinfectants. We did a paper on TWiM, if you remember, about how microbes have become resistant to hand gels, the common hand sanitizers.
Yeah.
And so consequently, I think a lot of, if microbes are anything, they're very good at organizing their thoughts and controlling resistance. They're putting it all into a package that can say, do I need it today or can I leave that luggage at home? And they obviously brought this luggage to space with them for a reason. And my guess is it's conferring a selective advantage to the microbes to live in space.
Which is a concern.
A lot of the antimicrobial resistance genes, remember, are dealing with our immune system, which throws out free radicals. And space has naturally more free radicals because of the no ozone to filter the UV, cosmic rays, which are splitting water and making free radical oxygen moieties inside the water of the cells. And so there's a lot of things going on that was probably driving the AMR operon to stay put, because we don't know what's all in those AMR operons from the perspective of dealing with oxidative stress, which is our principal way our immune system deals with infections. We cause oxidative stress in the bacterium. They give up the ghost and throw up their hands and they're dead. And we principally do it through the generation of peroxide and free radicals.
They also find viruses, which are, of course,
not surprising,
human origin, all kinds of polyoma, papilloma viruses, herpes viruses of all different sorts. And as they said, you get frequent reactivation of latent herpes viruses in space because of the stress conditions. And so those end up on environmental surfaces and also swabs from them as well. The one thing that I thought was, and that's the last data point, is interesting. they look at the phylogenetic distance between bacteria on the ISS and on the Earth. This is something that Mark Martin referred to last week, which he wished they could have done on the China space station. But here we have isolates from Earth and from the space station, and they can see that there's some adaptation going on. The sequences are different, but the antimicrobial resistance is the same profile of genes on the space station and on Earth. So, except for one bacterium where the antimicrobial genes show a distinct clustering on the space station compared with Earth. So they're going to evolve eventually everywhere, right?
Well, that was the one microbe that I found peculiar was the Panoptera pearsonii, which has only been isolated in space. Right. But when you look at its phylogeny, it's a derivative of Erwinia, which is a plant pathogen, which I'm certain are on plants on Earth. And it was probably brought with their fresh vegetables that they get each time it goes up. And then the selective pressure of the harsh space environment selected out. So that's how they were able to isolate what they thought was an innocuous microbe. But now we know that this new gram negative is a mesophile and it can cause disease in people.
And I'd like to give a shout out to just how thorough this study was. They looked at over 31 million 16S sequences, 3.7 billion metagenomic reads, and nearly 21,000 chemical features. They point out that this is 100 times larger data set than those collected previously. They also have a really thoughtful discussion about, so this is what we've seen. How should we be thinking about it, feeling about it, planning for it? Much like hospitals have a higher load of antibiotics and therefore antibiotic-resistant microbes, they're concerned that all the use of the sanitation bits to keep everything clean there is going to, of course, put us at risk of having microbes there that are now resistant to these detergents. Right. So they're going to continue this sampling. And they point out that we might want to consider probiotic-based sanitation measures, which is interesting to me. Haven't considered that.
Yeah, they say that the data can help inform, changes to the space station that could make it better for human habitation, and also to minimize. There's a lot of cross-contamination between the modules, and they said they would like to minimize that. Obviously, you don't want the toilet module to be contaminating other places as well. So these are actionable data, I think.
Right. And again, instead of finding like scary new microbes that are from space, instead, I think one of the surprising takeaways for me is I didn't appreciate that many of the astronauts are having rashes, et cetera, things like that may be due to absence of the missing microbes that are promoting good health. So be careful what you wish for if you keep it too clean.
When space exploration began, we didn't know anything about beneficial
nothing.
Microbes.
Right.
60s, right? I mean, nothing. And then long term, nothing at all. So now we know, and we have to deal with it if you want to stay up there.
Yep.
Okay. Now we're going to turn to pain, Michael.
We're going to turn to pain. This is a paper from a journal that we normally don't interact with. This paper is from Neuron, and it's an open access paper. It's going to be published July 9th, 2025, but you can download it presently. And it's from a group of 40 authors. Kai is the first author, and one of the corresponding authors is Amir Mindby. And they are principally from the Ministère de la Santé et des Services Sociaux, or the Ministry of Health and Social Services of Quebec, and the Technion, or the Israeli Institute of Technology, and then, of course, McGill University, which is also in Montreal, Quebec. And as listeners of TWiM can attest, if you write a provocative title, you might catch my eye. Well, the 40 authors did indeed catch my eye with their title, The Gut Microbiota Promotes Pain in Fibromyalgia. Amazing. And this is a fascinating paper But the in brief summary provided by the journal Neuron sealed the deal and had me into reading this fascinating paper. And you have to be brave to read a Neuron paper. This paper is 53 pages long. So I pity the poor undergraduate who has to summarize this for our ongoing TWiM activities. But I'm going to help you out a little bit and quote a little bit of the in brief and let you be the judge if you would dive into it. Kai et al. Revealed that transplanting gut microbiota from women with fibromyalgia, a chronic widespread pain condition of unknown etiology, into mice induces pain. Okay, so far. I can now imagine what sort of experiments that I might want to propose to address this hypothesis. So I feel comfortable going into the meat of the paper. But the next sentence, it also induces phenotypes commonly observed in patients. So now I got the translational science piece of it. This is real in people, such as immune activation, an outcome I can measure, metabolomic changes, another outcome that is easily measured, and reduced skin innervation. Okay, I was good with everything from that sentence until I stumbled into wondering, do I know the meaning of the phrase reduced skin innervation? You know, when I went to college, there was no such thing as neuroscience. So I looked it up and learned that skin innervation refers to the network of nerves supplying the skin, providing both sensory information and controlling autonomic functions like temperature, regulation, and sweating. I learned it as the network includes afferent or sensory and efferent autonomic neurons with a dense distribution across all layers of the skin. And then looking further, I discovered an open access paper that I'm going to drop into the show notes for you from 2019 in the Annals of Neurology by Ebnikoff that offered that distinct patterns of skin denervation in patients with fibromyalgia syndrome show a spectrum from a normal to generalized reduction of intraepidermal nerve fibers, which is reflective of severity of the fibromyalgia symptoms. So fewer nerve fibers, greater pain. So I'm now ready to go into the meat of the paper. But wait, I need to learn a little bit more about fibromyalgia, which I knew just a surface level from talking to my rheumatology colleagues and attending seminars. This is a mysterious condition characterized by chronic, and I underscore the word chronic, widespread pain. And I underscore the word pain because these poor souls can barely get out of bed, can barely move because of the pain. They also have fatigue and impaired sleep. And it's extremely challenging to diagnose, often where the primary care team says, it's all in your mind. And then they send them out to a psychiatrist or they prescribe an antidepressant. And they end up showing up at my rheumatology colleagues and they shake their heads and they say, we just don't know what triggers it, but it's real.
They don't have any injuries, right? There's nothing
going on.
Yeah, that's the
So
problem.
they don't have a way to treat it either.
No. And fibromyalgia affects up to 4% of the global population. That's 320 million people and occurs in the absence of tissue damage. In 2019, one of the corresponding authors, Mindbree, and his colleagues discovered that gut microbiomes of women with fibromyalgia differed significantly from those of healthy women. And so I'll drop that into the show notes because, again, another open access paper. So if you know someone who's suffering from it or you're interested in it, you can read up on it. So I'm back on track with the authors. And in fact, that is the central hypothesis of this paper. Could a dose of microbes from healthy people ease the pain and fatigue caused by this illness of unknown etiology? After all, previous research that we've covered on TWiM has shown that gut microbes might indirectly influence an array of chemical signals tied to the perception of pain. And in fact, another systematic review published in 2024 is entitled implication of intestinal microbiota in the etiopathogenesis of fibromyalgia. And this is, again, a systematic review. And as Michelle will tell you, I have a tendency to fall through the looking glass. But I was glad that I did here, in that I learned that in fibromyalgia patients, quantitative imbalances of the intestinal microbiota that we all know as microbiologists as dysbiosis, and their bacterial metabolites, those short-chain fatty acids with differential relative abundances, have been found, especially with the short-chain fatty acids in LPS, and that the microbiota has been found to indirectly influence host neurotransmitter mechanisms, mainly through, wait for it, the serotonin precursor, glutamate and gamma-aminobutyric acid, which we previously covered on an episode of TWiM, albeit was concerned with salmonella. So I was now ready to see if these authors would be able to generate the data that their proposed mechanism of dysbiosis is indeed a contributor of this pathogenesis of fibromyalgia. Well, up front, you can tell this is a sell price paper. 40 authors and a lot of data-packed figures. What their paper offers, though, is a 2-for-1 special. In the first part of the paper, they do indeed show that fecal microbiota transplantation from fibromyalgia patients, but not from healthy controls, into germ-free or notobiotic mice, does indeed induce pain. And also, all of those things we could measure, numerous molecular phenotypes that parallel known changes that are observed in fibromyalgia patients. So we're looking at mice.
Could I
Please.
emphasize this? They have several quantitative assays. They're not asking the mice how they're feeling. This is quantitative physiology, and it is quite convincing.
I mean, this isn't made up. The mice aren't lying, folks.
Mice can't tell you anything.
Well, they can jump off of hot plates and you can measure it. And that's one of their tests. And what they did is they replaced, or if you will, swapped out the fibromyalgia microbiota with healthy microbiota, and that substantially alleviated the pain in the mouse. The second aspect of their manuscript is a goosebump raising set of observations for those suffering from fibromyalgia. Here they offer us data from an open label, albeit small pilot trial in women with fibromyalgia that showed that transplantation of a healthy microbiota is associated with reduced pain and improved quality of life. Now, granted, there were only 12 patients that got better out of 14, but that's a big number, folks.
And if I could just interject here, the first author, Huihua Kai, got in touch, and she said it was just an extraordinary day when her advisor, Dr. Arkady, told them that Dr. Amir Minerbai was going to do this clinical trial. She said, "Oh my gosh, now my lab research is actually has the potential to have real world impact." And she felt a deep sense of fulfillment and responsibility to do these experiments and to continue this path.
Yeah, it's really phenomenal. And this was a clinical trial. There's a registry number that you can look up. But let's go back to the data where they're beginning to tease apart the mechanism, because that's what we really want to understand as a microbiologist. In their first set of experiments, they present data that the gut microbiota transplantation from fibromyalgia patients was able to induce pain. And the experiment was pretty straightforward. They collected
in mice, right?
In mice. When I get to people, I'll tell you it's people. They collected the fecal samples from women with fibromyalgia in an equivalent age and health matched healthy control. And then they transplanted them into this germ-free mouse model or notobiotic mice. The interventional stool, or the fibromyalgic stool, was collected from donors suffering from primary fibromyalgia. Simply, it was the only thing they had, no other comorbidities. They screened negative for anxiety, depression, irritable bowel syndrome, or other comorbidities that can disrupt the gut microbiome, like, for example, diabetes. you can check out the inclusion and exclusion criteria that the authors provide in the supplementary content. And this, again, is an important and critical control in all of these studies. These authors were meticulous in making sure that all their I's were dotted and T's crossed. Now, the microbiome transplantation was done weekly and was performed by transplanting human stool from women with fibromyalgia and age-matched healthy controls into the germ-free mice. And that's, you can see in figure 1. And I'm going to skip over the microbial characterization because on TWiM, we've seen those methods before, and they're simply offering us controls differ from the intervention. No big surprise because they had previously published that, and you can go and check that out. But figure 1E, they begin to use metagenomic data where they observe substantial differences in bacterial communities between the two groups, similar to those observed using the 16S. The next set of their experiments are the behavioral studies, where they actually demonstrate pain of the mice getting either the fibromyalgia microbiome or the control. And here they show that the fibromyalgia transplant from the individuals of fibromyalgia, but not the healthy controls, into germ-free mouse induced the development of mechanical hypersensitivity in this von Frey test. And I don't have enough time to explain that test, but you can Google it. It's a standard test used in neuroscience. And in fact, our listeners who are taking neuroscience probably can recite it chapter and verse. They also looked at thermal hypersensitivity in the radiant heat paw withdrawal test, cold hypersensitivity using a cold plate, spontaneous pain measured via the mouse grimace scale, and the muscle hypersensitivity by applying pressure and application measurements. And these are all quantitative that you can measure over and over again with great reproducibility. And what they learned is, yes, indeed, folks, there is a difference between the fibromyalgia microbiome and that of the healthy control. And this chronic widespread pain in fibromyalgic patients is often accompanied by cognitive symptoms and depression. Now, here, they transplanted the fibromyalgia gut microbiota, and the mouse that got the intervention stool did not show any deficits in cognitive functions. And as you can guess, it's a rat maze or a mouse maze. And so it didn't impact their cognition. They also assessed metabolic change in mice colonized with the intervention microbiome using targeted metabolomic analyses. And again, not surprisingly, found that mice that received the fecal transplant from the fibromyalgia patients exhibit an altered metabolic profile compared to their control. And you don't even have to read the paper too deeply because the figures are so easy to look at. The fibromyalgia is the colored one. And whatever they're measuring, it's opposite of what the control is doing. So it's really easy to see. Then they also assessed immune alterations in the mice colonized with fibromyalgia microbiome. And here again, not surprisingly, and this is through analysis of the immune landscape conducted using single-cell RNA sequencing of peripheral blood mononuclear cells, or PBMCs. It again reveals that the mouse colonized with the interventional stool had an increased proportion of classical monocytes compared with mice that received the healthy control. Remember, more monocytes means not good. And again, analysis of the differentially expressed genes within this population showed enrichment for IL-17 and tumor necrosis factor. Again, you don't have to have much immunology to know these are the danger signals which are associated with inflammatory responses.
And Michael, could I add that they did not see this pathology within hours of the transplant? It's not like they were adding a toxin that was now going all over the mouse body. It took weeks. So this is having an impact, as we're going to, they'll suggest, on pain receptors. And
right.
Their neurobiology.
Which is why I was glad I fell down that rabbit hole and looked at this whole issue of nerve intervention, because I don't know anything about neuroscience beyond what I hear in seminars. And here's where the colonization of germ-free mice from individuals with the interventional stool not only induced changes in the immune system, but they saw this increase in peripheral monocytes and reactive spinal microglia. This is also consistent with their observation of low-grade peripheral inflammation, and they also detected increased excitability of the dorsal root ganglia neurons that are the first neurons in the sensory pathway. They too can contribute to this pain hypersensitivity. So they're building us a mechanism of how whatever the microbiome from this fibromyalgia cohort is conferring to the metabolism, and the mice are all eating the same rat chow. It's just that the microbes processing the mouse chow are different. And they then also went down the path to investigate whether disruptions to the intestinal barrier or immune cell homeostasis, which they thought may account for the fibromyalgia phenotype. And they did this by conducting histomorphological and immunophenotyping analysis, where, again, they sack mice, they look at the histology to see if the microbes are effectively getting into the system that could explain it. And they didn't see any changes in intestinal integrity. So it's suggesting that the microbes are doing some signaling, but they also saw an increase in IL-17 production that may be attributed to the heightened activation of the gamma delta and CD8 positive T cells, which to me, again, is not that surprising given that these two populations can be substantially influenced by microbial composition, at least from what we've been covering on TWiM. They also offer another fascinating hypothesis that it's this absence of gut damage along with the limited immune response and unchanged visceral sensitivity that suggests that the fibromyalgia gut microbiota contribute to peripheral phenotypes. And this sort of blew my mind, such as peripheral immune responses, metabolomic alterations, pain, and muscle hypersensitivity via distinct mechanisms that still are unknown, rather than through the commonly accepted or often bandied about hypothesis of gut barrier dysfunction or just straight up inflammation.
There's probably some soluble mediators that are made in the gut and travel everywhere else, right?
Well,
they can go to other organs.
They refer to some literature that says it's the dysregulation of bile, which I thought was just detergent that helps us digest food. Turns out it can bind receptors and trigger different modulation of immune function, and also
that was one of the most convincing set of experiments. These mice that were transplanted with the interventional stool from the fibromyalgic patients showed reduced levels of specific bile acids. So this is why it's important to look at everything. And it was the ursocolate and the bile acid metabolizing bacterium, Lactoclostridium sedendens. It used to be Clostridium sedendens. It's now the evil taxonomist renamed it to Lactoclostridium. They treated these mice with UDCA, which is a host-derived primary bile acid metabolized in the mouse gut into various secondary bile acids. And the pain hypersensitivity in the mouse was partially alleviated, supporting that the absence of these good bugs is responsible, or dysregulated bile acid metabolisms may be one of the fundamental triggers underlying pain in fibromyalgia. And bile acids through the receptors, and again, this is classic neuroscience, G protein coupled, but this is now a bile acid receptor, the 5-TGR5, and the farsinoid X receptor, FXR, exert numerous effects on host physiology, including the modulation of immune function. I'm sure this is old news to the folks in neuroscience, but it was news to me. And it's because of the microglia maturation function. And what it does is promotes analgesia, or pain reduction. Finally, they found a decrease in the intraepidermal intervention of the fibromyalgia recipient mice. And as I offered in the opening, this reduced density of intraepidermal fibers has been extensively reported in a subset of individuals with fibromyalgia. And this is people, and have been linked to immunologic and metabolic disturbances. Although the underlying mechanisms in functional role in mediating pain and other fibromyalgic phenotypes in people are, as you might guess, not well understood. So summing up this half of the paper, fibromyalgia is complicated. No one will disagree. The authors go to great lengths in helping us, the naive reader, demystifying the pathogenesis of this disease that may be simply as straightforward as one who has fibromyalgia may simply be suffering from dysbiosis of their gut.
Bad
Michael,
bugs.
So is it, you say dysbiosis, so it's an altered composition. Is it
Yes.
missing something? It's missing something that keeps others in check, maybe?
It's the community. You may not have a number of the right players there. And I'm sure the human race doesn't eat mouse chow. So it's going to be much harder to do based on our diet. Yeah. And the one thing the only criticism I had is we don't have genetics of the people. We have genetics of the mice. The mice are all the same.
And to the author's credit, they point out that this pilot study they did in humans, which got incredibly exciting results, is just
which I'm just about to
convincing
get
that we
to.
need to continue to pursue this line of investigation because there's great promise here.
But we could use fecal transplants like we do for C. diff sometimes.
Oh, yeah,
right?
absolutely.
Yeah.
But that is easy. That's easy stuff. You just want to eliminate one creature. Here, we're literally reinterpreting the opera.
Yeah.
We're putting a mezzo soprano in where there was a bass, or we're putting
we don't know yet. We don't know.
We don't know
how many microbes you'd have to change. But let's we're running low on time, so let's get into the human data.
This gets us to figure 6, where the title of the figure gives you the idea where many of the patients suffering from this disease would like every one of our labs to go to. Move into the open label clinical trial of healthy control fecal microbiome transplantation in women with fibromyalgia. That's right. They're testing the hypothesis. If we restore balance, and since I found this paper on the 4th of May, can we restore balance to this life force? After all, Rita Colwell, the former National Science Foundation director, often in her remarks at the Centennial General Meeting of the American Society of Microbiology, that the midichlorians, the microscopic sensitive life form that lives symbiotically within the cells of all living beings, are essentially for connecting with the force. Now, that's science fiction from Star Wars. But what we're effectively doing is we're asking if we can simply restore balance to the fibromyalgic gut. Will things get better? Now, figure 6 is a primer. And apologies to Vic DeRita. He doesn't like when I use that word, but apologies to Vic. Offers a schematic representation of this pilot study. Panel A gives you what they've done. Panel B is the consolidated standard of reporting trials diagram that is ubiquitous in every clinical trial. To folks new to clinical trials, it's a ubiquitous figure that you must find in every clinical trial where you are reporting randomized controlled trials. Simply put, Panel B offers how many participants were enrolled, how many were randomized, how many completed the study, and why some participants were excluded.
And let me also add that in order to transplant a different gut microbiota to these willing volunteers, they swallowed poop pills, basically. They were encapsulated in a pill. So it wasn't anything more disgusting than that.
No lavage. And in fact, fecal transplantation has gone a long way since that seminal paper in the New England Journal that demonstrated that you could disrupt C. difficile diarrhea with a stool transplant. Panel C is pain intensity. And again, it looks very similar to the same sorts of experiments that they did in the mice, but they have literally quantitative measures and which they looked at everything. So just look at figure 6. Bottom lining this was an open label pilot study where 14 women on average who were 52 years of age, plus or minus 13.7 years, with severe fibromyalgia and refractory fibromyalgia were enrolled. The question they asked, did the healthy donor stool from a similarly matched 3-person donor pool do the trick? In a word, the pilot was small, the data were insufficient to draw definitive conclusions, but as you can witness for yourself, that in panel 6C, all the way through the rest of those panels, that 1 week after the last healthy control transplant, pain and intensity decreased by greater than 2 points out of 10. That's phenomenal. 12 of the 14 patients resulted in a clinically and statistically significant decrease in the mean pain intensity. As you again follow through panels E and I, the systematic burden of fibromyalgia, you can tell the systematic burden decrease and anxiety, depression, and sleep quality and physical quality of life scores improve, which is the hope of every patient suffering from fibromyalgia. They did mental quality of life scores showing a trend, and you can again see it's working. In closing, this is but the tip of the iceberg. This is the topic of the gut microbiome. As you longtime listeners will know, it has many twists and turns. This team is now working on a larger clinical trial that would enroll 80 participants and include a control group. The senior author, Mindbee, thinks that future clinical trials will help identify the specific bacteria responsible for fibromyalgia-related pain. These organisms could be replaced or removed or engineered not to do something. And the final piece of information I'd like to leave with you is a quote from one of the senior authors of the paper that came from one of the Nature News articles I wrote. And this is Amir Minerby. For years, we've not offered patients any effective treatments, and the medical system has disregarded their symptoms. We really owe them. And this paper is a guiding light, the light at the end of the tunnel that, good science will have dividends.
So this fecal transplant in these people, it seems after the treatment, it seems the pain seems to start going up again. So
it's not a stable community. We don't yet know how to engineer stability. Mm-hmm. In the case of Clostridium difficile, it's really simple because you eliminate that organism. And that's what the stool transplant does.
That's a toxin-mediated disease, unlike this, which gets into the nerve system.
This is metabolism. You're eating food. And we have to make certain that the community is right. And we're only beginning to scratch the surface on how to get the right communities.
And the right bile composition, right?
That's it. Yeah.
Which is encouraging. So the first author of this paper is a postdoc at McGill. This is Weihua Kai. They did their bachelor's and PhD degree in China and have been studying chronic pain for years. I think the first publication was in 2016 on this topic. While they were doing their graduate work, they had the opportunity to go to Rutgers for a couple of years and work closely with a lab there that is doing in the Department of Anesthesiology that was also doing pain-related studies. But now as a postdoc in Arkada's Kortowski's lab, she has just been so grateful for his mentorship, who encouraged her to think independently, lead the project, stay positive and resilient, but more importantly, taught her the true meaning of collaboration. It's not just working side by side, you do this, I'll do that, but actively talking to each other, seeking advice. And building a network. This is what gives you the power to tackle complex problems. So she is her advice for aspiring scientists. She turns to a Chinese saying whose translation is, when everyone adds fuel, the flames rise high. And it certainly has in this. So she interprets that by saying science truly is a team sport. Support each other, the brighter your discoveries will shine, the more where you work and collaborate. Now, when her experiments don't go as planned, she's able to turn to playing the piano. Music is her favorite way to relax, stay positive, clear her head. She's also very grateful for her supportive family, who's been really encouraging her all along. And she also is especially grateful for her supportive husband and their lively 16-month-old daughter, who adds a bit of chaos, but also brings endless joy to their lives. So congratulations on this fantastic study. And thank you for sharing your perspectives with us.
It's amazing to get first authorship on this paper, right? Because there's so many authors.
Uh-huh.
Yeah.
She must be very happy.
Yeah, and she has multiple first author papers just in the last couple years, including some in eLife, and she's also published on pain in Science, Frontiers in Cellular and Developmental Biology. So, been working with some really productive collaborations and leading a number of them.
Leadership, that is the key. That's great. Thank you, Michelle.
Yeah,
thank
team
you,
science.
Michael.
I like
that
it.
That is TWiM 332. You can find show notes at microbe.tv/TWiM. You can send any questions or comments to twim@microbe.tv. And if you enjoyed these programs, please support us. Microbe.tv/contribute. Michelle Swanson is at the University of Michigan. Thank you, Michelle.
Thank you. Great papers, you guys.
Michael Schmitz at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. See you next time on This Week in Microbiology.